Good day, everyone. I'm Krisa Maikidis, and I will discuss the study of microbial growth. When microbes are provided with nutrients and the required environmental factors, they become metabolically active and grow. Growth takes place on two levels. On one level, a cell synthesizes new cell components and increases its size. On the other level, the number of cells in the population increases. This capacity for multiplication, increasing the size of the population by cell division, has tremendous importance in microbial control, infectious disease, and biotechnology. In the following sections, we will focus primarily on the characteristics of bacterial growth that are generally representative of a single-celled microorganisms. The objectives. Compare and contrast the phase of growth in a bacterial growth curve. The basis of population growth. Binary fission. The division of a bacterial cell occurs mainly through binary or transverse. Fission, binary means that one cell becomes two and transverse refers to the division plane forming across the width of the cell. During binary fission, the parent cell enlarges, duplicates its chromosome, and forms a central transverse septum that divides the cell into two daughter cells. This process is repeated at intervals by each new daughter cell in turn, and with each successive round of division, the population increases. The stages in this continuous process are shown in greater detail in Figure 7.13 and 7.14. Figure 7.13 shows the steps in binary fission of a rod-shaped bacterium. The first step in binary fission is a young cell at early phase of cycle. The second is a parent cell prepares for division by enlarging its cell wall, cell membrane, and overall volume. Midway in the cell, the wall develops notches that will eventually form the transverse septum, and the duplicated chromosome becomes affixed to a special membrane site. The third step is the septum wall grows inward, and the chromosomes are pulled toward opposite cell ends as the membrane enlarges. Other cytoplasmic components are distributed randomly to the two developing cells. The fourth one is the septum is synthesized completely through the cell center, and the cell membrane patches itself so that there are two separate cell chambers. And the last one, at this point, the daughter cells are divided. Some species will separate completely as shown here, while others will remain attached, forming chains or doublets, for example. The rate of population growth. The time required for a complete fission cycle from parent cell to two new daughter cells is called the generation or doubling time. The term generation has a similar meaning as it does in humans. It is the period between an individual's birth and the time of producing offspring. In bacteria, each new fission cycle or generation increases the population by a factor of 2 or doubles it. Thus, the initial parent stage consists of one cell, the first generation consists of two cells, the second 4, the third 8, then 16, 32, 64, and so on. As long as the environment remains favorable, this doubling effect can continue at a constant rate. With the passing of each generation, the population will double over and over again. The length of the generation time is a measure of the growth rate of an organism. Compared with the growth rates of most other living things, bacteria are notoriously rapid. The average generation time is 30 to 60 minutes under optimum conditions. The shortest generation Times average 5 to 10 minutes, and longer generation times require days. For example, Mycobacterium leprae, the cause of Hansen's disease, has a generation time of 10 to 30 days as long as some animals. Environmental bacteria commonly have generation time measured in months. Most pathogens have relatively short doubling times. Salmonella enteritidis and Staphylococcus aureus, bacteria that cause foodborne illness, double in 20 to 30 minutes, which is why leaving food at room temperature, even for a short period, has caused many persons to be suddenly stricken with an attack of foodborne disease. In a few hours, a population of this bacteria can easily grow from small number of cells to several million. Figure 7.14 shows the mathematics of population growth. The first graph is about the starting with a single cell if each product of reproduction goes on to divide by binary fission. The population doubles with each new cell division or generation. This process can be represented by logarithms or 2 raised to an exponent or by simple numbers. In the second graph, it shows plotting the logarithm of the cells produces a straight line indicative of exponential growth, whereas plotting the cell numbers arithmetically gives a curved slope. Figure 7.14 shows several quantitative characteristics of a growth. The cell population size can be represented by the number 2 with an exponent 2 raised to 1, 2 raised to 2, 2 raised to 3, and 2 raised to 4. The exponent increases by 1 in each generation, and the number of the exponent is also the number of the generation. This growth pattern is termed exponential. Because these populations often contain very large number of cells, it is useful to express them by means of exponents or logarithms. The data from a growing bacterial population are graphed by plotting the number of cells as a function of time. The cell number can be represented logarithmically or arithmetically. Plotting the logarithm number over time provides a straight line indicative of exponential growth. Plotting the data arithmetically gives a constantly curved slope. In general, logarithmic graphs are preferred because an accurate cell number is easier to read, especially during early growth phases. Predicting the number of cells that will arise during a long growth period, or yielding millions of cells, is based on a relatively simple concept. One could use the method of addition: 2 plus 2 equals 4, 4 plus 4 equals 8, 8 plus 8 equals 16, 16 plus 16 equals 32, and so on, or a method of multiplication. For example, 2 raised to 5 is equals to 2 times 2 times 2 times 2. But it is easy to see that for 20 or 30 generations, this calculation could be very long. An easier way to calculate the size of a population over time is to use an equation such as NF is equals to Ni 2 raised to N. In this equation, NF is the total number of cells in population at some point in the growth phase. Ni is the starting number, the exponent N denotes the generation number, and 2 raised to n represents the numbers of cells in the generation. If we know any two of the values, the other values can be calculated. Now let us use the example of Staphylococcus aureus to calculate how many cells will be present in an egg salad sandwich after it sits in a warm car for 4 hours. We will assume that Ni is 10, or number of cells deposited in the sandwich while it was being represented. To drive N, we need to divide 4 hours or 240 minutes by the generation time. We will use 20 minutes. This calculation comes out to 12, so 2 raised to n is equals to 2 raised to 12. Using a calculator, we find that 2 raised to 12 is 4096. Final number, or NF, equals 10 times 4096 equals 40,160 bacterial cells in the sandwich. This same equation with modification is used to determine the generation time, a more complex calculation that requires knowing the number of cells at the beginning and end of a growth period. Such data are obtained through actual testing by a method discussed in the following section. The population growth curve. In reality, a population of bacteria does not maintain its potential growth rate and does not double endlessly, because in most systems, numerous factors prevent the cells from continuously dividing at their maximum rate. Quantitative laboratory studies indicate that a population typically displays a predictable pattern or growth curve over time. The method traditionally used to observe the population growth pattern is a variable count technique in which the total number of live cells is counted over a given time period. In brief, this method entails: first is placing a tiny number of cells into a sterile liquid medium. Second is incubating this culture over a period of several hours. The third step is the sampling the broth at regular intervals during incubation. The fourth one is plating each sample onto solid media, and the last one is counting the number of colonies present after incubation. Stages in the normal growth curve. Data from an entire growth period 3 to 4 days typically produce a curve with series of phases termed the log phase, the exponential growth, or log phase, the stationary phase, and the death phase, shown in figure 7.15. The log phase is relatively flat period on the graph when the population appears not to be growing or is growing at less than the exponential rate. Growth lags primarily because the newly inoculated cells require a period of adjustment, enlargement, and synthesis. The cells are not yet multiplying at their maximum rate, and the population of cells too sparse or dilute that the sampling misses them. The cells reach the maximum rate of the cell division during the exponential growth, or logarithmic or log phase, during which the curve increases geometry. The length of the log period varies somewhat from one population to another. It is important to note that even though the population of cells is not increasing or growing, individual cells are metabolically active as they increase their contents and prepare to divide. At the stage 8, at the stationary growth phase, the population enters several modes in which cells stop growing or grow slowly. The curve levels off because the rate of cell inhibition or deals balance out rate of multiplication. The decline in the growth rate is caused by depleted nutrients and oxygen, plus excretion of organic acids and other biochemical pollutants in the growth medium due to the increased density of cells. As the limiting factors intensify, cells begin to die at an exponential rate, or literally perishing in their own waste, and they are unable to multiply. The curve now dips downward as the death phase begins. The speed with which death occurs depends on the relative resistance of the species and how toxic the conditions are, but it is usually slower than the exponential growth phase. Viable cells often remain many weeks and months after this phase has begun. In the laboratory, refrigeration is used to slow the progression of the death phase so that cultures will remain viable as long as possible. Figure 7.15 shows the growth curve in a bacterial culture. It shows the stages in the normal growth curve: the log phase, exponential growth phase, and stationary phase, or the rate of cell growth equals rate of cell death caused by depleted nutrients and oxygen, excretion of organic acids and pollutants. Steps in viable plate count based culture method. A growing population is established by inoculating a flask containing a known quantity of sterile liquid medium with a few cells of a pure culture. The flask is incubated at that bacterium's optimum temperature and timed. The population size at any point in the growth cycle is quantified by removing a tiny measured sample of the culture from the growth chamber and plating it out on a solid medium to develop isolated colonies. This procedure is repeated at evenly spaced intervals. For example, every hour for 24 hours. Evaluating the samples involves a common and important principle in microbiology. One colony on the plate represents one cell or colony forming unit or CFU from the original sample. Because the CFU of some bacteria is actually composed of several cells, consider the clustered arrangement of Staphylococcus, for instance. Using a colony count can underestimate the exact population size to an extent. This is not a serious problem because in such bacteria, the CFU is the smallest unit of colony formation and dispersal. Multiplication of the number of colonies in a single sample by the container's volume gives a fair estimate of the total population size or number of cells at any given point. The growth curve is determined by graphing the number for each sample in sequence for the whole incubation period. Because of the scarcity of cells in the early stages of growth, some samples can give a zero reading even if there are viable cells in the culture. The sampling itself can remove enough viable cells to alter the tabulation, but since the purpose is to compare relative trends in growth, these factors do not significantly change the overall pattern. Turbidity measurements as indicators of growth. The first picture is about holding a broth to the light is one method of checking for gross difference in cloudiness. The second one is the eye is not sensitive enough to pick up fine degrees in turbidity. More sensitive measurements can be made with a spectrophotometer. A tube with no growth will allow light to easily pass. Therefore, more light will reach the photodetector and give a higher transmittance value. In a tube with growth, the cells scatter the light, resulting in less light reaching the photodetector and therefore gives a lower transmittance value. Growth patterns in microorganisms can account for the stages of infection. A person shedding bacteria in the early and middle stages of an infection is more likely to spread it to others than is a person in the late stages. The course of an infection is also influenced by the relatively faster rate of multiplication of the microbe, which can overwhelm the slower growth rate of the host's own cellular defenses. Understanding the stages of cell growth is crucial for work with cultures. Sometimes a culture that has reached the stationary phase is incubated under the mistaken impression that enough nutrients are present for the culture to multiply. In most cases, it is unwise to continue incubating a culture beyond the stationary phase because doing so will reduce the number of viable cells and the culture could die out completely. It is also preferable to do stains, or an exception is the spore stain, and motility test on young cultures because the cells will show their natural size and correct reaction, and motile cells will have functioning flagella. For certain research or industrial application, closed batch culturing with its four phases is inefficient. The alternative is an automatic growth chamber called the chemostat or continuous culture system. This device can admit a steady stream of new nutrients and siphon off used media and old bacterial cells, thereby stabilizing the growth rate and cell number. The chemostat is very similar to the industrial fermenters used to produce vitamins and antibiotics. It has the advantage of maintaining the culture in a biochemically active state and preventing it from entering the death phase. Other methods of analyzing population growth. Microbiologists have developed several alternative ways of analyzing bacterial growth qualitatively and quantitatively. One of the simplest methods for estimating the size of a population is through turbidometry. This technique relies on the simple observation that a tube of clear nutrient solution becomes cloudy or turbid as microbes grow in it. In general, the greater the turbidity, the larger the population size, which can be measured by means of sensitive instruments. Enumeration of bacteria. Turbidity readings are useful for evaluating relative amounts of growth. But if a more quantitative evaluation is required, the variable colony count described in Insight 7.6 or some other enumeration or counting procedure is necessary. The direct or total cell count counting the number of cells in a sample microscopically in Figure 7.17. This technique very similar to that used in blood cell counts, employs a special microscope slide or cytometer calibrated to accept a tiny sample that is spread over a pre-measured grid. The cell count from a cytometer can be used to estimate the total number of cells in a larger sample, for instance, of milk or water. One inherent inaccuracy in this method, as well as in spectrophotometry, is that no distinction can be made between dead and live cells, both of which are included in the count. Counting can be automated by sensitive devices such as the culture counter, which electronically scans a culture as it passes. Figure 7.17 shows the direct microscopic count of bacteria, a small placed on the grid under a cover glass. Individual cells, both living and dead, are counted. This number can be used to calculate the total count of a sample. Electronic counters, such as culture counter, microbial suspension forced through a small hole or orifice. Movement of microbe through orifice impacts electric current that flows through orifice. Cannot distinguish living from dead cells. Quick and easy to use, useful for large microorganisms and blood cells but not prokaryotes. It is being adapted for use as a rapid method to identify pathogens in patient specimens and to differentiate blood cells. More sophisticated forms of the flow cytometer can actually sort cells of different types into separate compartments of a collecting device. Although flow cytometry can be used to count bacteria in natural samples without the need for culturing them, it requires fluorescent labeling of the cells you are interested in detecting, which is not always possible. A variation of the polymerase chain reaction or PCR, called real-time PCR, allows scientists to quantify bacteria and other microorganisms that are present in environmental or tissue samples without isolating them and without culturing them. To summarize this lesson is these are the chapter summary with key terms. The splitting of a parent bacterial cell to form a pair of similar-sized daughter cells is known as binary or transverse fission. The duration of each division is called the generation or doubling time. A population theoretically doubles with each generation, so the growth rate is exponential, and each cycle increases in geometric progression. A growth curve is a graphic representation of a closed population over time. Plotting a curve requires an estimate of live cells called a viable count. The initial flat period of the curve is called the lag phase, followed by the exponential growth phase in which viable cells increase in logarithmic progression. Adverse environmental conditions combine to inhibit the growth rate, causing a plateau or stationary growth phase. In the death phase, nutrient depletion and waste buildup cause increased cell death. Cell numbers can be counted directly by a microscope counting chamber, culture counter, flow cytometer, or newer methods that detect organisms in their natural settings. Cell growth can also be determined by turbidometry and a total cell count.